My name's Paul Brown. I'm an Associate Director for Pharmacology and Toxicology in the Office of New Drugs in the Center for Drug Evaluation and Research at the Food and Drug Administration. And my task is to talk about the regulatory framework for assessment of carcinogenicity of human pharmaceuticals. So I think a good place to start with this discussion is to talk about the statute language that governs the regulation of new drugs and biologic products, as well as the Code of Federal Regulations. So the Food and Drug Cosmetic Act governs the regulation of new drugs, while the Public Health Service Act governs the regulation of biologic products. And both of these statutes are fairly general in their language and don't really mention carcinogenicity specifically. They talk about the need for information about the safe use of a drug without getting into details. The Code of Federal Regulations for the regulation of new drugs, as well as for biologic products, gets into a little more detail. The content and format section of the regulations talk about how there's a need for pharmacology and toxicology information as appropriate to support the safety of a drug for topics such as carcinogenicity. The section of the regulations to talk about biologic license applications is, again, fairly general and just talks about safety. I'm underlined as appropriate because not every drug is going to require a carcinogenicity assessment, and I'll talk a little bit about that in the subsequent slides. Since the statute and regulations don't really provide much detail, what we do rely on are guidance documents. So we create guidance documents largely through the International Council for Harmonization process, and that's abbreviated ICH. And the ICH S1 guidance documents are specifically focused on carcinogenicity assessment. And there's also a number of other guidance documents through ICH that can touch on carcinogenicity assessment. For example, for biologic products, the ICH S6 guidance document, and for small molecules, the ICH M3 guidance document. So ICH S1A is a good place to start. It talks about the need for long-term rodent carcinogenicity studies, and it goes through a number of considerations for when you might need to do a carcinogenicity assessment for a drug. So it considers things such as if treatment duration is 6 months or greater, then generally a carcinogenicity assessment is going to be needed, or if the use is intermittent for a chronic indication. That's another situation where carcinogenicity assessment is likely to be needed. There may be other cases where even if the treatment duration isn't 6 months or longer, that carcinogenicity assessment might be needed. For example, if there's a cause for concern, maybe there's pre-neoplastic findings in earlier toxicity studies, or if the compound's genotoxic, those might be reasons to do a carcinogenicity assessment, even if the duration is relatively short. On the other hand, there may be considerations about patient population or the indication that would consider that the carcinogenicity assessment isn't needed. So for example, if the life expectancy of the patients is relatively short, a carcinogenicity assessment might not be needed. So once you go through ICH S1A and you determine that a carcinogenicity assessment is needed, then you would move on to the other guidance documents. So for example, I already mentioned ICH S6 for biologic products. In that, it talks about doing a carcinogenicity assessment primarily based on a weight of evidence type of approach, where information about the biologic product and its target and the subsequent biology is analyzed and discussed in terms of its impact on carcinogenicity. And this data can come from a variety of sources. In many cases, a carcinogenicity study in rodents isn't feasible with a biologic product because many of these biologic products are so specific for their target in humans that the rodent isn't a relevant pharmacologic species. In addition, sometimes these biologic products cause an increased immunogenicity in the animal models, and you really can't do a long-term rodent carcinogenicity study. There are rare cases where we don't understand enough about the biology and a rodent study is feasible, where a carcinogenicity study might be conducted with biologics. But as I say, that's relatively uncommon. If you have a small molecule and you need a carcinogenicity assessment, then you would consult ICH S1B guidance documents, the S1B and the S1B R1 addendum, which is a relatively recent addendum that's come out a couple of years ago. And in this approach, it describes in ICH S1B the need for one long-term rodent carcinogenicity study and then an additional carcinogenicity study, either another long-term study or perhaps a shorter or medium-term study, such as a 6-month study in a transgenic animal. The ICH S1B addendum has provided a weight of evidence assessment approach to determine whether a 2-year rat study adds value to the overall assessment. So it that document talks about providing that weight of evidence along with a 2-year or a 6-month study in mouse. The ICHS-1C guidance document talks about dose selection for carcinogenicity studies. And so once a study is determined to be needed, you could consult that guidance document on guidance on how to select doses for the carcinogenicity study. This is a flow scheme from the ICHS-1B addendum that talks about the weight of evidence approach. So you can see that sponsors would gather the information for all the factors to consider in that guidance document, and the next slide goes over those factors, and then conduct an integrated analysis of those factors and determine one of 3 possible outcomes. So on the left is an outcome where the carcinogenicity potential in humans is considered to be likely. So in that case, you wouldn't likely need to do another carcinogenicity study to confirm that, and that would be potentially accepted as an argument that a 2-year rat study or even a mouse study might not be needed. In the middle is the outcome where the carcinogenic potential is considered to be unlikely in humans, such that a 2-year rat study wouldn't add value. And in that case, the sponsor would provide that weight of assessment to the regulatory authorities, and there would be a determination of whether that was acceptable. And if so, then a 2-year rat study wouldn't be necessary. But in general, a mouse carcinogenicity study would still be an expected part of the assessment. Or the sponsor may find on the right-hand side that the carcinogenic potential in humans is uncertain. Perhaps there's not enough known about the pathway and its carcinogenic potential. In that case, the approach generally outlined in ICHS-1B would be followed, which is the long-term rodent study and an additional study. These are the factors that are listed in the ICHS-1B for the weight of evidence assessment. You can see them listed in the middle of the slide. It includes target biology, secondary pharmacology, histopathology from chronic studies. And since this is a determination of whether a 2-year rat study is going to add value, that chronic toxicity study histopathology data is going to come from a 6-month rat study generally. And then there's also an assessment of hormonal effects, whether the compound's genotoxic, and whether it has any effects on immune system or immune suppression or other modulation. And so the approach would be that the sponsor would have information to address each of these factors, and they would weigh that information to determine whether it supports it is less likely to need a 2-year rat study or more likely to need a 2-year rat study. So for example, if you look at target biology, if this target is a well understood target, perhaps there's other molecules, drugs in the class that affect this target, then there's a good understanding of what the biology of the pathways are and their relationship to carcinogenicity, well, then that would support the conclusion that a 2-year rat study is less likely to be needed. However, if it's a new pathway, not a lot of good understanding of the biology, perhaps not a good understanding of how it relates to carcinogenicity potential, then that might lead you to conclude that a 2-year rat study could add value and it therefore is more likely. And then you would do that process through each of these factors and come up with an overall weight of evidence as to whether the study is likely to add value or not. You can note that in the bottom of the slide, this also allows for the introduction of other types of information into this consideration. So it lists things like in vitro data and emerging technologies. So the overall procedure, I've sort of discussed this already, for sponsors is to conduct that assessment on all the factors described, determine if this does support the conclusion that a 2-year rat study would not add value, and if so, submit that assessment to the application with a request that the assessment be accepted in lieu of a 2-year rat study. I'll note that this assessment and this need to not conduct a rat study isn't really considered a waiver. We are still assessing the carcinogenicity according to the regulations, but it's just an assessment that doesn't really require a rat study. As far as within the agency, these requests for weight of evidence assessment are submitted to an application. Review division would review that, and then it would evaluate the sponsor submission and present the sponsor's rationale and the division's recommendation to an internal committee. That committee would then discuss it and come up with a conclusion and a recommendation for the division, and then the review division would then communicate the agreement or disagreement with that sponsor's proposal back to the sponsor. I'll note that there is a triage process built into this such that if that information isn't available in the weight of evidence submission, the information that I listed for each of those factors, including that chronic toxicity study, then there is a possibility that the review division would reject it and not review it until that information is provided. We've been doing this now for a little over a year, and so we have some experience with what makes a good quality weight of evidence submission. And perhaps obvious, but still important, is just the organization of the submission. That helps the review division as well as the committee that's reviewing this find the information and be able to reach conclusions. It's useful if the application discusses each weight of evidence factor and its relationship to carcinogenic pathways and the potential risk associated with those. The application should be transparent and unbiased in its presentation of findings, both from the literature as well as the toxicity studies, and address those findings in terms of their relevance and impact. And even though we need all that information, it's also helpful if the application succinctly explains why the data from the 2-year rat study would not add value to the overall carcinogenicity assessment. In many cases, a carcinogenicity study is still going to be needed. Even when there's a weight of evidence assessment, there's the expectation for the mouse study. And we've found that about 75% of the time, there's still going to be a need for a rat study as well. So the agency does accept protocols from carcinogenicity studies for review ahead of time before the studies are conducted. And those can be submitted under what's known as a special protocol assessment. And there's a guidance document that explains that process. And then we also have a guidance document specifically for carcinogenicity study protocol submissions that lays out the kind of information that we need to see in order to do these reviews. So a special protocol assessment is mostly used for clinical trial protocols, but as I say, animal carcinogenicity studies are eligible for a special protocol assessment. This is a 45-day review period. The sponsor should include all the relevant background information in addition to the complete protocol for the carcinogenicity study in the request. Some of the important information that we need to review to support a protocol include toxicology dose range finding study reports. Those are generally 90-day studies to support a 2-year carcinogenicity study, but they can be of shorter duration for an alternative bioassay. For example, a 1-month dose range finding study is sufficient to support dose selection for a 6-month transgenic mouse study. It's also useful to have metabolic profiles from humans and the species employed in the tox assessment, as well as TK data and AUC data for the parent and major metabolites, so that we can make sure that the parent and major metabolites are adequately covered in the proposed carcinogenicity study. It's useful to have plasma protein binding data as well, so that we can determine whether it's appropriate to use the total drug or the free drug levels for these comparisons. And then it's useful to have the gene tox profile as well. The special protocol assessment may not be appropriate if it contains a request to evaluate more than one protocol. We like each protocol to be a separate special protocol assessment. It may not be appropriate if it contains a protocol for an ongoing study. We don't do a protocol assessment for a study that's already been started, or if it does not contain sufficient content and detail. So all that information I just listed, we need to have in the special protocol assessment request, or else we can't do the assessment, and those can be rejected before we even begin to look at them. So the process is similar to the weight of evidence assessment I talked about. The request for special protocol assessment will go to a review division. The staff will review it, and then they'll bring that to an internal committee. In this case, it's called the Executive Carcinogenicity Assessment Committee. And that committee will determine if the protocol is acceptable. Concurrence with the general protocol design and the doses is documented in writing, and that constitutes a special protocol agreement. If the committee can't agree with the sponsor's proposed protocol design, but there is adequate information in it to determine doses and determine an appropriate protocol design, then the committee may make recommendations based on that. And if that's followed by the sponsor, then that also constitutes an SPA agreement. There can be cases where the exec CAC does not agree with the proposed protocol design and can't come up with a recommendation, and that would be considered a non-agreement. If the sponsor gets a non-agreement, they can resubmit the request with additional information to address the deficiencies, or they can continue and conduct a carcinogenicity study without a formal FDA agreement. A special protocol assessment agreement isn't required in order for a study to be conducted. So once the studies are conducted and completed, how are they evaluated for human pharmaceuticals by the agency? Well, the carcinogenicity studies are generally submitted at a time of marketing application, so a new drug application or a biologic license application, again, unless there's cause for concern. So for example, similar to what I mentioned earlier in determining when a study is even necessary, if there were pre-neoplastic lesions, for example, in the toxicity studies, or if there's a clinical sign for carcinogenic concern, or if the compound's genotoxic, these may all be reasons for conducting a carcinogenicity study earlier in development and submitting it earlier than marketing application. All the study data are submitted electronically to CDER, and that includes individual animal data and includes outcomes such as mortality, body weight, and then the neoplastic and non-neoplastic findings. All that information is reviewed in detail by a pharmacology toxicology reviewer as well as by a statistical reviewer. And the mortality is statistically analyzed, as are tumor trend and pairwise analyses corrected for mortality. And then there's a center-wide conclusion by that executive carcinogenicity assessment committee on whether there are any drug-related tumors in that study. So once the conclusion is reached as whether there are drug-related tumors, that can be considered in the overall risk-benefit analysis of the pharmaceutical. So we ask questions like, are tumors relevant to the patient population? What's the margin between the human exposure and the exposure causing the tumors, or the exposure not causing the tumors? What's the indication in patient population? Is this for a serious disease or an unmet medical need, in which case acceptable risk might be higher? Or is this primarily for a pediatric population, in which case the acceptable risk might be lower? So after that analysis, there can be several outcomes from that risk-benefit analysis. It could potentially impact the approval of the drug if the risk is considered unacceptable, or it could lead to things like altering the dose or patient population. For example, you could limit the duration of treatment if you have a concern, or you could potentially exclude patients with a history of malignancy. Another approach could be to increase monitoring of patients. If the carcinogenicity study results in a tumor in the animals for which there's an analogous human tumor, you might be able to monitor for that tumor in patients more frequently, for example. Regardless of whether the study is considered positive or negative for findings, it will be described in the labeling, so it will be available there for healthcare practitioners and patients to make decisions. The last couple slides talk about some new areas of interest in the assessment of carcinogenicity of human pharmaceuticals. We think the ICH S1B weight of evidence approach provides the opportunity to bring some new methodologies to bear on the evaluation of carcinogenicity potential. And we think this could be true for pretty much all the factors within that weight of evidence assessment, such as the primary pharmacology or target biology, or the secondary pharmacology. There are a lot of different molecular targets that can be screened in secondary pharmacology. So I think we want to help explore what molecular targets add the greatest value for informing carcinogenicity potential. One of the assessments that's conducted as part of the weight of evidence is whether there's any hormonal perturbation. And so I think we can ask the question, are there reliable translatable methods to predict in vivo presence and absence of hormonal effects? And likewise with immunotoxicity, are there reliable translatable methods to predict in vivo presence and absence of immune effects that may be related to carcinogenicity? And then finally, we think that the weight of evidence to support a conclusion that a drug is unlikely to be carcinogenic in humans, the data for that need to be reliable at predicting no concern. This might be a little different from using some of these sorts of data for other types of evaluations. So an in vitro screen with a high sensitivity to detect a potential carcinogenic signal, but a low specificity may not be particularly useful for this approach. But we do think that methods that address things like key characteristics of carcinogens, or that assess the impact of a drug on expression of cancer driver genes or other carcinogenomic endpoints, may be valuable additions to the weight of evidence assessments. And then we want to ask questions like, which of these methods would be most impactful? Which could be the best to start using first? Which are available and which are affordable? So I hope this summary of the approach to framework for assessing human pharmaceuticals for carcinogenicity and some of the opportunities for future assessment will be helpful in your considerations. Thank you.